ठीक है, फिर मिलते हैं हम लोग। Okay, so let's get started. Okay, so yeah, let's get started. I believe you all have your own computer. Okay, so there are 5 questions. Go through one question, starting with the first question. Do each question and let me know that you are finished. Okay, if you are having difficulty, I am going to help you. I will come to you also to discuss as you are working on this. So the first question is, let me read actually the first question. The first question is, so why this is this is not working, this side. Okay, so I am going to read question number 1. So what you will do, you want to get the genome information for bacterium Pseudomonas aeruginosa. Okay, you will visit the NCBI website to extract this information. So go to NCBI. If you have opened your computer, go to NCBI. Google search NCBI or just type NCBI in the address tab. In particular, extract the genomic data for the strain Pseudomonas aeruginosa PA1. This is the model Pseudomonas aeruginosa. What is the size of the complete genome of this strain? Okay. So as you are going through this data, you will get lot of information about this genome. I am asking you to get the size of the complete genome of this strain. Okay. The next question is, how many protein coding genes are there in the genome of this organism? So there could be both non-coding RNA genes and protein coding RNA genes. So typically when people say a gene, in general it means gene that codes for a protein. But there are genes that do not code for proteins. They could be RNA genes. So only RNA transcripts will be formed that will not get translated to proteins. So you have to get the number of genes that code for proteins in this genome. So that's why protein coding genes. So yeah, be careful about this. And then the next question is, P. aeruginosa PA1 is known to be pathogenic. What disease is this pathogen associated with? So you have to get all this information from the database. Identify a virulence associated gene in this pathogen. Provide the name of this gene. Gets its coordinate in the genome. Coordinate means a start and end position of the gene in the genome. It's a start and stop codon and the size of the protein it encodes. Next question is, obtain the 15 nucleotides of this genome. So you will get to the genome page and look for the last 15 nucleotides. Note that the genome may be circular. I am asking you to provide the last 15 nucleotides as in the FASTA sequence of the genome. Okay. Is it a part of a protein coding gene? So you have to look at the annotations, so the features in this genome. So that should help you answer this question whether this is a part of a protein coding gene or not. Assuming no information about this sequence except the order of nucleotides, translate it into an amino acid sequence using a standard genetic code table. Start with the first 3 nucleotides as the first codon and so on. Stop translation when you encounter a stop codon. Okay. So get started. Go to NCBI and look for Pseudomonas aeruginosa PA01 genome. Able to get there? Not yet. Okay. Okay. Pseudomonas aeruginosa. So, yeah, okay. I think you should type Pseudomonas aeruginosa and then not genome. The question is genome. Actually, let's go to the question. Okay, so genome. So yeah, scroll down here. Yeah, it's okay. Type it. Genome. Yeah, and then type Pseudomonas mo aeruginosa. I think GI PA1 PA. Yes. Okay. Yeah, it is. So now I think you can go to the next step. Okay, how is it going? Genome, yes, Pseudomonas aeruginosa. Mm-hmm. Mm-hmm. So how is it? Oh, you got it? Okay, okay. Then the question is that once you have found the genome, question is what is the size of this genome? Okay, so can you look for the size? I think that is also provided. So you are there. Yeah, right. Good. So go in this page. You should be able to find the size of this genome. So look for that. There is some problem with the okay. Network issue. Oh. Hmm. Yes. Oh, yeah, you got it. Yeah. So, what is the size of the genome? Yeah. So, if you, I think it will. So, you see, this is the exact 6 to yeah, 6,264,404, right? Yes. 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 Yeah. Good. Yeah. If you put the cursor here, then you will get the exact. Okay. So, note down that. Note down. Got it? No, no, you got it. Click on this. Yeah, that was the accession number, and go down, see the genome size, right? Yeah, good. You got this? Yeah, yeah. Scroll down. Oh, you're in taxonomy, and why not start with the genome actually? So go to, yeah, maybe here, or you can from NCBI genome. I think here also. Okay, can you? Yeah. Oh, yeah. So you, yeah, go to this. Actually, the first one is PO1, the reference genome. Click on that. Yes. Okay, good. Scroll down. See, yeah, genome size. If you bring the cursor here, then it shows the exact size. Yeah, right. Are you able to do? Yeah. Yeah, protein coding. Yeah, genes, right? Yeah, that's total. Actually, above that is total genes, and below that is only protein coding. Yes, right, right. So yeah, now go. Now you have the right place. So go through and try to answer these questions. Going well? Okay, good. So where you are? Oh, third question. Okay, okay. So you got assembly, and this is the size. So yeah, if you so annotation detail. So annotation is here, right? So this is total gene number of genes, and this is only protein coding. So yes. Okay, I think it's going on well. Um so it seems like most of you or all of you have answered, except those having some internet problem. Uh, so far as the genome size is concerned and the number of protein coding genes in this genome. The third question is that this strain is known to cause diseases. What disease is this pathogen associated, or what diseases? There are several diseases. Uh, so that information you can also get from the genome site. Look for papers uh, published on this. The original paper on PO1, I think it was published in 1990. You can check actually the complete genome of Pseudomonas aeruginosa PO1, um, published in journal Nature. Okay. So you should be able to get that paper. I think it is public, that paper, and that should give you information about the diseases this pathogen is associated with. Okay. So this is a genome paper um, published in Nature in I think 90s something. So check for this paper, the complete genome of 2000 or 2000, the complete genome of Pseudomonas aeruginosa PA1 published in year 2000. Okay. Yeah. So if you go through that, read it, and you will see the mention of diseases associated with PO1 strain, and also it should help you in identifying uh, genes that are associated with virulence. So yeah, try it. So are you able to? Okay. Okay, so yeah, you're good actually. So this is a reference string. So click on this. Yeah, click on this accession, uh, and go down. Yeah, go down. Yeah, so this is the genome size. So if you move the cursor here, you can see the exact size. No, yeah, yeah, that is also, yeah, that's okay, yeah. Yeah, but this is total ungapped. It's better to move cursor. I think both are same. This is right. Yeah, so both are same. Total size for total number of genes. You have to you look for annotation. So protein coding genes. Yeah, here you can see total genes and protein coding genes. Okay. Okay, so are you all able to open the Pseudomonas aeruginosa PA1 paper, original paper, published in Nature? Can you see mention of the diseases associated with this strain? You are able to, yeah. For example, which disease? Urinary tract infection. Okay, okay. Yeah, there are many diseases. Yeah, it is often mentioned in the context of cystic fibrosis. Yeah. Okay, so yeah, keep trying. Yeah, let me know if you are getting stuck. Yeah, you have to identify a virulence associated gene in this pathogen. So a gene that is responsible for pathogenicity in this organism, which is involved in pathogenicity. So virulence associated gene. Uh, you can find information of virulence factors in the original paper, the Pseudomonas aeruginosa PA1, and then you can look for those genes in the genome database. Okay, so make sure that when you open the genome page, select all features. Okay, let me. So I think most of you are able to get to the genome of this strain. So this one actually, which is the reference. So if you click on this, then you get information about this genome. So genome size is 6.3 MB. If you put cursor there, you can see the exact genome size. And then at the bottom, you will see the annotation details. So total number of genes is 5,697, and out of this, 5,572 are coding for proteins. So the protein coding genes is actually this 5,572. Okay, so you were able to do this, and then you went down and then clicked on the reference genome, right? RefSeq. So when, and you can see here also the genome size. So when you click on this, you will go to gene bank page. So here is the gene bank page. Due to large size of this record sequence and annotated features are not shown. But you want to, so from this actually you can find the original paper. So I am looking for that. and I think it is at the top. Oh, here this one. Complete genome sequence of Pseudomonas aeruginosa PA1, an opportunistic pathogen. So if you will click on this, so let's do new tab. Okay, so here is the paper, and this paper is now publicly available. So click on this, and then read it. Actually, in the main or later, it should mention about the diseases caused by this pathogen. So it does mention bacteremia in burn patients, urinary tract infections, hospital acquired pneumonia, cystic fibrosis, and so on. So many different diseases caused by this pathogen. Okay, so which one? Oh, okay, yeah. I actually I look for okay, see this actually. This article actually complete genome sequence of Pseudomonas aeruginosa PA1, an opportunistic pathogen, published in Nature in 2000. Okay, and I clicked on the PubMed ID because I wanted to get access to this paper. Okay. Okay, and then I clicked on Nature portfolio, which is on the right top of this page. And then I could get access to this paper. And in this paper, it mentions in the first paragraph of the main section the diseases caused by this pathogen. Okay. Okay, and then you can look for virulence factors in this genome. So maybe a search actually, virulence or pathogenicity. So find on this page virulence. Okay, so virulence factors. Okay, the are biosynthetic genes. Exoenzymes and systems that secrete them. Okay, so okay, let's go to more specific information. Regulation of virulence. Okay, so yes. Oh, you got it for virulence factors. Yes. Oh, another paper. That's fine. So in the original paper also it should mention in export of extracellular virulence. No, not export. We want to get virulence factor genes. So protein secretion. Okay, here it is. P. aeruginosa secretes several virulence factors, including toxins, lipases, and proteases. Okay, and it should also provide the names of some of these genes. So let's consider toxin, and I'm going to search for toxin in this gene bank file. But because this is large, it has not been shown here, but you can customize view. So click on customize view, and then select customize. Okay, and then all features and update view. So it will download then all features, all genes. So you have to go to customize and then select all features and then update view. So it's a large file, I think about 20 MB. But this will get done in a few minutes. And then we will look for a toxin gene in this. Okay, done. And then when you scroll down, then you will see all the different features, the genes which are present in this genome. And if you go to the bottom, you will also see the genome, I think, or maybe not. That's fine. But it is showing you all the features. But when you select download sequences, so if you go to customize again and then show sequence, then it will show you also the sequence. Okay. So, and I want to look for toxin. So I will search here toxin. Okay, so here it is, exotoxin A. Okay. So, and this is a virulence factor. So, what is the question actually? So, go to the question. So, identify a virulence associated gene in this genome, which you did. Okay. So, exotoxin. Provide the name of this gene, which you did. Get its coordinate in the genome. What is the start and end position of this gene in the genome? So, and then look for this gene actually, which is this one. And the start position is this, end position is this, and this is on the complementary strand. Okay. Okay. So, and CDS, I think they should be the same. Okay. What is the next question? It's a start and a stop. Now do this actually. Find its a start and a stop codon. Okay. So, we are here, and show me that you have found the start and stop codon of this gene. If you are looking at exotoxin, if you are looking at some other gene, that's also fine, but get me the start and stop codon of the gene that you have identified, the virulence factor gene. So could you? Oh, it is still
not responding.
It is not
Responding.
Oh, Gaurav, yeah, many of the students, they are having internet problem. So, okay, could you find a start and stop codon?
No.
Not yet,
Yeah,
but you are there.
just,
Is this okay? So this is where it says exotoxin. So, no, here it is, right? Product is exotoxin. Okay. So, okay, you have got the coordinates. You have to get the start and stop codon of this gene. Okay. So, start and stop codon, you will want to click on CDS. Did you find? Yeah. Okay. Good. So then click on faster. So you're here. Click on faster. Okay. So start ATG and go to the end. Stop is TAA. Yeah. Very good. So you got it. So start is it. Oh, sorry. Sorry. And stop is very good. Yeah. Did you get this? Okay. So you are here. This is exotoxin.
yes, I found that.
Okay.
I found the
Uh,
genome coordinate too.
Okay, so this is tox A. Okay, and then this is the coordinate, and what it says, product name class 1, exotoxin. Okay, click on CDS because you have to get the start and stop codon, and then go down, and yes, here, and click on faster. Okay, so start codon is ATG, and stop codon is TAA. Okay. Start and stop codon. Got it? What is the start codon? And stop codon? Got it? Oh, no, yeah, but this is not the right actually. So go to and then look for a toxin. Exotoxin, yes. So all features, did you do all features? Okay. No, 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 no. Go back, and Pseudomonas aeruginosa, right? Can you scroll down and show me what it is? Okay. Select at the top. Select all features. Okay. So customize view. Customize. Yeah, I think why it is not giving. Okay, go down. Uh, yeah, okay. Go down actually. I want to. Yeah, you are seeing all these features. Now look for, yeah, search exotoxin. Oh, it is still downloading. Okay. Yeah, exotoxin. Oh, yeah, but these are. Yeah, you have to go to Pseudomonas aeruginosa web page and then. So yes, yes. Click on this. This taxonomy, but I think from here also go down, and then no, yeah, yeah. Here is the genome actually. This, click on this RefSeq genome. This. Yeah, RefSeq, yeah, reference, and then yes, right. Click on this. Yeah, here it is, and then go down and look for go down. Okay, here it is RefSeq. This is the accession number. Click on this. Click on this, and then select customize view. Customize all features, and then update view. So then all features will come actually. So all genes, and then look for exotoxin gene. Okay. Okay, here it is, and you want to get the start and stop codon. So click on CDS, coding sequence. Okay, coding sequence. Okay, it is not clicking. Yeah, so click. Oh. No, actually, I don't know what happened. Go back, or maybe it did actually, because it is showing. Go down then. Oh, go to again exotoxin. Oh, okay, yeah. Click on CDS. So okay, yeah, here it is. And click on FASTA at the bottom right. No, no, FASTA at the, yeah, below FASTA. Yeah.
Recently, several variants of the toxin like this.
Yeah, toxin. Yeah, you got toxin actually. So then go to.
Why you search for exotoxin specifically when we are doing then why how we know that which word we search and then we click on CTS. You have आपने कहा कि एक्सोटॉक्सिन पे क्लिक करना है तो आप कैसे एक्सोटॉक्सिन पता चलेगा कि एक्सोटॉक्सिन यहीं पे ही क्लिक करना है
नहीं वो वहां से जाएंगे
नहीं वो तो पता है
पता हो गया
एक्सोटॉक्सिन में क्यों आप मतलब बोल रहे हैं उसको
ओ क्यों
पूछ रहे हैं
ओ क्योंकि नहीं, इसमें टॉक्सिन हम ढूंढ रहे हैं, तो एक ही सर्च आ रहा है एक्सोटॉक्सिन,
अच्छा
तो इसीलिए जो है
अच्छा टॉक्सिन
कर रहे हैं तो वो एक ही जीन दिख रहा है, तो ये दिस इज, या इसीलिए अब सीडीएस पे क्लिक कीजिए,
हाँ वो तो करिए
सीडीएस पे, हाँ यस, एटीजी राइट, एंड देन स्टॉप इज yes, yeah, this is, and then this is the coordinate, and then you want to get the start and stop codon, so click on CDS. Okay, and then FASTA here. Okay, so here it is. Start codon is ATG, and stop codon you will have to go to the end. And in DAA. Okay. Okay, so it seems like you were able to get the start and stop codon. So for that you clicked on CDS, and then at the bottom right you clicked on FASTA, and here is the FASTA sequence of exotoxin gene coding sequence. So first codon ATG, which is the start codon, and then last codon is TAA, which is a stop codon. Okay. Okay, and then let's look at the next question. The size of the protein it encodes. Okay, so I think some of you might have already got it, but this you can get from the same page. Go back, and then this is CDS, and it may mention about protein. So can you see protein somewhere? Protein ID. So click on protein ID, and then you will see the number of amino acids. So 638 amino acids. This is the size of the protein. Okay. Yeah, I did this for you. But this is, are you able to do it? Got it. So, okay, very good. 638, I mean, this is the size of the protein this gene codes for. Okay. The next question. Next question is, obtain the last 15 nucleotides of the genome. So, you will have to download this genome. We'll go back to the page, genome page, and we are here. So I think we can do from here. We can just say source sequence, okay, and then update view. So let's see actually. So now our sequences would also be downloaded. So download the sequence which I am doing because we want to obtain the last 15 nucleotides of this genome. So you will have to download the genome. Okay, so it's a large sequence. Okay, complete. So I will go to the end. Okay, I will go to the end. So here is the genome actually in the end. Can you see the have you downloaded? Because I just again I went to customize view and I also selected source sequence. So it has downloaded the sequence. Same thing actually. Now you have to also check on source sequence. So it will download the sequence. And then you have the genome sequence downloaded, and you have to get the last 15. So this is actually shown in every 10 actually. So if you see the position here, 6,264,361. This is the coordinate of this nucleotide T. And this is I think 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is 10, 10. Okay. So, and then this would be 370. This is 361, this position. So, this would be 371, right? And this would be, so I am not saying 6264, so you understand. So, this is 361, this is 371, this is 381, this is 391. Okay. So, 391. And from here to here, how many nucleotides? This is 10 and this is 4. So, 14. So, then you will have to include this also. So, the last 15 nucleotides would be this, right? Can you do this? If you have not, then do this. Even if you understand, repeat it. Do yourself. Go to the same gene bank page. Customize view, select all features, and then select source sequence, and then update view. Okay, you should have now the genome sequence also downloaded. Okay, and go to the last and get the last 15 nucleotides, and which I found to be this one: GTC, GCA, GTG, ACC, CCG. Okay. Sravani, could you do it? Okay, go to the genome, Gaurav.
Genome?
Yeah, genome page. Pseudomonas aeruginosa, PO1. Type of PAO1. Okay, those who have downloaded that page, actually this page, try to do the next question. Okay, the question is, I think, to check whether this is part of a protein coding gene or not. So do that and let me know. Okay. Okay, so did you get it? Okay, genome. Yeah, very good. So yeah, this is the reference genome. So select this one, the assembly. Okay, and then go down, and you will get the accession for ref-seq. Click on this ref-seq, and yeah. So due to the large size, it is not downloading all features. So you will have to select customize view and allow it to download everything. Customize all features, sequence also, and then update view. It will download the genome sequence. So could you answer the next question? Is it part of a protein coding gene or not? Yes.
I have this master file of this here,
Yeah.
but this is only.
No, I think, yeah, this is some gene actually. You have to go to the genome. Go back.
It's a complete genome.
No, okay, complete genome. I don't know, because that's not, because this is just about 2000 base pair. So I think it is not the right. Yeah, this is just showing the region. So this is a large genome. Hmm. Okay. Okay, so if you look at all features and then look for the last feature, the last gene, which is this gene. Okay. So which is starting at this position and ending at this position. And last 15 nucleotides, you know the coordinate of that. Yes, because the last nucleotide has the same coordinate as the genome size, right? So, so you know the coordinate of the last 15 nucleotides, and then you will check whether that is overlapping with this or not. So, what is the size of this genome? So, yeah, take a, let's go to. Okay, okay, I will have to do again this. That's fine. So this is the last. The last nucleotide is located at this is size of the genome, right? Okay. So 6264404. So remember 6264404, and I will go again to the last. Okay, okay, here. Okay, so this is 6264361. This is not right. This is not. So this last, because it ends here, and this is the last feature. So clear? So last 15 nucleotides is not part of a protein coding gene, because that was 6264404. Yes, 4404. So 406. Okay, clear? So you have to get the coordinates of the last 15 and see whether this is overlapping or not with the last feature, and we see that it is not, because last feature is ending at 4 million. So this, oh sorry, 6,264,361. So last 15 nucleotides, they come after this. They come after the last feature, the last gene. So last 15 nucleotides, then they are not part of a protein coding gene because it ends, that is the last gene. Clear? Okay. So now use the genetic code table, and I think I have provided you lecture 1 genetic code table. So okay, not in this. Okay, let me open BHU. And yeah, standard genetic code table, if you just Google search also, you will get it. NCBI also has, I showed you that. So if you go to NCBI taxonomy, you will get the genetic code. So which is here. So you are going to use the standard genetic code, this one, to translate the last 15 nucleotides to protein sequence. Okay. So you have the last 15 nucleotides. You're downloading. Oh, you got last 15 nucleotides? No? Downloading. So if you go back, go up. Oh, hanging. Okay. Oh, it disappeared. Oh, yeah. Let it download. Let it download. Uh, it is not downloading. So can you go down actually towards the end? No. So yeah. This from this actually this, and then yeah, and okay, pull it down. Last. Okay. Oh, the download has been cancelled. I don't know what this is happening.
This is the fifth time we downloaded.
Yeah. Fifth time it didn't happen. Okay. But did you understand? Okay. Okay. Could it download? Okay. So you are here, and then select on also sequence. Then you will get the sequence. So show sequence, and then update view. And then once done, you go to the bottom and get the last 15. Okay. Okay. So are you translating last 15 nucleotides to protein? Done. Okay. So okay, so let me get to that. Okay, show me what you did. So GTC 1, 2, 3, 4, 5. Valine, alanine, valine, 3. Okay, so I don't know actually, but I will trust that this is correct. Okay. So same. Oh, so this validates. Okay. Okay, good. So Sravani, could you? Oh, it is still downloading, and Gaurav, you can.
Still downloading and
Oh, it is still downloading.
Yes.
10.87 MB.
Oh, this is really slow.
5 to 6% use my data, so
Oh, okay. Okay, it will go about 20 or 22, and then we'll stop. Done?
last one is loading for proline.
Yes, right. And so first is
Okay.
no. So first, yeah, this is GTC is the first. So you have to GTC.
Yeah, I was working for CG.
No, yeah, yeah. So can you write actually the 15 GT beginning with GTC? GTC. Okay, so the first one is, so what GTC codes for? So you will write that, and then next is GCA, what it codes for. So write actually those amino acids. Yes.
I'm searching for this.
Oh, you are.
I have written.
So first one is valine. Oh, okay. So GTC codes for valine. Okay, good. And next is
Next is alanine.
alanine. Okay. So next is, you can check actually. Next is after GTC GCA. Yeah, yeah. GCA, what it codes for. Okay, it seems like there is some download problem also. Uh, so we are done with this actually. I took about 1 hour. Okay, so let's look at the next question. Yeah, while you all are working on this, and keep working on this, it may take more time, that it's okay. Yes.
आफ्टर डाउनलोडिंग शो किधर होगा? हमारे डाउनलोड को शो नहीं होगा, उनका हो गया है, मेरा नहीं हुआ।
Oh, downloaded.
Yes, sir.
Oh, download and then go down. Okay, can you? It should show. Yes. Go towards the end. It is still downloading. So yeah, but it is about to complete.
Mine is downloaded, but where it will show after
Yeah, genome will show at the end.
at the end.
Yeah.
When it's showing off
like that.
Yes, like this. Yeah, it is showing. So it is.
series.
Yeah, same. It will be same. Can you show Gaurav? And I think Sravani, your is complete. So go down, and will be the genome sequence. Okay, wait. Yeah, it is. Go down. Yeah, I think it's memory problem also likely. Yeah, I think memory problem. So yeah, I think there is some internet speed problem as well as memory problem with your computer. Maybe your computer is not that powerful, which is okay, but you should be able to do it now that you understand. So let's go to next question. Okay. So try this. If you have not finished, we can discuss later on also, not a problem. But let's go, because we would want to go through all these questions by 4:30. So how many mouse proteins are smaller than 80,000 Daltons? How many mouse proteins are bigger than 80,000 Daltons? So can you go to NCBI protein and try to answer this question? Question number 2. Go to NCBI protein and then try to answer this question. Could you start? NCBI protein? Yes. Yeah, in the NCBI protein, you have to search for mouse proteins. So then mouse is organism. And then among those proteins, how many of them are smaller than 80,000 Daltons? So that is the molecular weight. Okay. And how many mouse proteins are bigger than 80,000 Daltons? So go to NCBI protein and look for proteins in mouse. NCBI protein. Yeah, but yeah, you should not do. Yeah, you should go to proteins. in data, or not all databases, proteins. Protein, yes, and then okay, then delete proteins actually, because you already did protein here. Protein, and type mouse, and then no, not yet. You have to write organism, because there could be sequence from some other organism, but somehow associated with mouse. So make sure that you choose only the proteins that are in mouse. Okay. So next question is mouse protein. So you have to go to NCBI protein, and then mouse. Okay, mouse. But I'm saying that you also mention organism, because if you don't, then it may pull some record which is of some other organisms, but somehow linked with mouse. I want you to get the proteins within some range of molecular weight only for this organism, mouse. So you will put organism, okay, and then click on this, and okay, so all these mouse proteins are here. So total 482,017. Okay. The question is, of them, how many are smaller than, so let's look at this. How many of them are smaller than 80,000 Daltons? Okay. So to do this, anyone has done this or not yet? How many of them are smaller than 80,000 Daltons? So of course you are going to type and all in uppercase, and because I am trying to do the next proteins, mouse proteins that are smaller than 80,000 Daltons. So and then from 0 to 80,000. So 0 to 80,000, 0 to 80,000. Okay. And what is this? Molecular weight. So you can type molecular weight or abbreviated mol weight. Okay. And then search. So 80,000, this search. Everyone, could you do this? Okay. So click on search. Oh, same. Okay. Okay. So we will do one thing. We'll refresh. We'll refresh this and do again. And 0 to 80,000, 80,000. Otherwise, we will do this by opening a new page. Yeah, what I will do is I will just, yeah, maybe same thing will happen, but I think I will try with a new page. Okay, so yeah, close this and go to, this is some server problem. So go to NCBI and then protein, and then this thing. Okay. Okay, because you all are hitting the same, so the server has become so busy. So yeah, internal error. So I think this is puzzling. Okay, so I will keep this open so that we will return to this later on. Okay. Which one?
Everything came and then it shows 0.
Where? Oh, no. You have to go to protein actually. So this.
I did it in protein.
Yeah, it's not protein actually. It's everything. Yeah, go back from here, right? And then select protein.
Okay, from here.
Yeah, from there. So protein and then type that. Okay, so we'll keep trying this actually. But in the meanwhile, let's look at the second part. How many mouse proteins are bigger than 80,000? So now your lower limit would be 80,000 and your upper limit will be some large number. So make it too large and then see how many you are getting. Make it still larger. At some point it will saturate. You will not start getting any more. So thus you will find the total number of mouse proteins with molecular weight 80,000 and above. Got it? Okay. So 80,000 and above, your lower limit would be 80,000 and upper limit would be some very large number. You will get something, increase that further. If you get the same number, it means that this is the total number of proteins with molecular weight greater than 80,000. Clear? Okay, so you will do it, and yeah, at some point I think we should be able to. For some reason, NCBI is not helping us get this value, and it doesn't matter if you type molecular weight. You should be getting the same, but it will still give you the server error, it seems like. Okay, so let's go to the next question. Use PubMed to search the research articles on von Willebrand disease. How many of them have phrase von Willebrand disease in title and abstract of the articles? How many of them are published in the journal Blood? So can you go to PubMed and try to answer this question? Question number 3. How much? How many? 229. Let me see. 229 in Blood. So, but how many are then in title and abstract? 19? 1963. Okay. Okay, so okay, so you have to go to PubMed and then advanced search, and then question is articles on von Willebrand disease, and how many of them have phrase in title? Okay, so this, and you can put this in double quote, but it should not matter actually. And then select title abstract, add, and then search. So 3,895. Okay, and then how many of these are published in journal Blood? So we'll go to advanced again, and then we'll add this, add query, and then we'll select journal. The journal is journal, and then which journal? Blood. Okay, Blood. Okay, so Blood. Okay, Blood, and then add. So this is end. So you want to find the number of papers with von Willebrand disease in title or abstract and published in Blood journal, and then search. How many? 229. Okay, so we did this. Okay, great. Okay, so next question, and again you have to search the NCBI database to find the accession number of von Willebrand gene in human. Okay, it is asking for accession number of von Willebrand factor gene in human. So you have to go to NCBI gene, and you will type von Willebrand factor and get me the ref-seq accession for this. Yeah, sometimes the servers are not able to access some specific database. That's why it can return error message. But you should be able to get the right response at some other time. Okay. So I think it is likely because the server is not able to access. So are you working on the fourth problem? Okay. So let's go to gene, and then this one, von Willebrand factor or VWF. And first try to get the accession for ref-seq accession for this gene, and then messenger RNA, and then protein. So yeah, gene, then mRNA, and then yeah, it should be not gene ID. You have to get the accession number. Gene ID would just be number. Accession number would be alphanumeric. Yes, if it is ref-seq, then it will start with N. Yeah, NC, or yeah, you did NG. Okay, you got it. Okay, okay. So I think yeah, it should be where. So this is, so click on this. Yeah, this is the right one, and then scroll down. Is it okay? It's yeah. If you will scroll down, then you will see ref-seq, gene, mRNA, and protein. Yeah, scroll down now. Yeah, scroll down. You should see ref-seq or more. Oh, here it is. Yeah, Rep6, and then this is for gene, this is for mRNA, and this is for protein. So these are the accession numbers. Okay. Okay. So I think you got it. So this is for the gene. So if you click on this VWF, then here it is, and then scroll down and look for Rep6, and then where it is, I did see actually variation. Here it is. Okay. Rep6, yes, reference Rep6 of annotated genomes. Okay. So here it is, but I think, yeah, this is the right one. So yeah, you can see the accession for VWF gene, von Willebrand factor gene. This is gene accession, and then the next one is mRNA accession, and this one is protein accession. These are Ref6 accessions. Okay, if you click on that, you will see the curated Ref6 sequence data. Good. Okay, repeat. Okay. Okay, so go to NCBI gene. So scroll down, scroll and see for gene. Okay. Okay, so NCBI gene, so which is here NCBI gene, and then type von Willebrand factor and then search. Okay. When you will do this, then it comes at the top. Von Willebrand factor, Homo sapiens human. Okay. Click on this. Click on VWF. Then it will give you all information about this gene, and we want to get accession numbers for gene, mRNA, and protein. And if you can get Ref6 accession numbers, then you would want to have that. So scroll down and you will see Ref6. So I see Ref6 here. Okay, Ref6, and here is the Ref6 accession number for this gene. NG underscore 009072.2. Okay, and then accession for mRNA is this, for protein it is this. Clear? Okay, and what is the next question now? Also the okay, we did this. Which chromosome is VWF situated in? 12. So we should be at the top actually. If you go to the top of this, then it should show you the information about. So here it is, location 12. So chromosome 12, which is also mentioned here, chromosome 12, and coordinates, which is the start position and end position, that is also mentioned. Okay. Okay. Next question. Search for coding sequence for this protein and provide the link to access the CDS. Now, can you do this question? Look for the coding sequence for this gene. So you have to get coding CDS for this gene. So you have got the accession numbers. Now look for CDS of this gene. Got it? Okay. Oh, right. So you have to click this actually, and then FASTA, and then download. Actually, it should be here. Yeah, this one. CDS. Is it CDS? Doesn't look like actually, because CDS will start with a start codon. So go to the accession, Rep6. Yeah, it will come at the end. No, I think go up. Rep6. No, yeah, just search for Rep6. Could you get it? Yeah, Rep6. You have got it. Yeah, Rep6. next. Yes, so you have these accessions. So click for you can click for gene bank. For this also, there you can see CDS. And now look for CDS. Yeah, look for CDS. CDS. Yeah, this. Okay, so you have to get the CDS coding sequence for this. So here it is. So these are the accessions. So this is for the gene, and CDS is within the gene. Okay. So you will click on gene bank for this, and then it will download the gene bank information. And then, so here is the gene bank page for this gene. You will go down and look for CDS. So gene, mRNA, exon, CDS. Okay. So CDS. So click on CDS, and then click on FASTA on the bottom right. Okay. And then okay. So here it is. So yeah, so it starts with ATG and ends with, let's see, yeah, it's a long CDS, ends with TGA. Yeah, many exons. This has so many exons. This gene. Okay. And then next question is search also OMIM and obtain the VWF accession number there. What is the function of this protein? So go to OMIM. Okay. Go to OMIM. So scroll down, OMIM, VWF, von Willebrand factor in human. Okay, which came at the top, von Willebrand factor, database is OMIM, and accession number is 613160. Okay. And what is the next question? Got the accession number? Okay, so now read this actually, read text, and let me know what is the function of this gene. Read the text description. What this gene does? Helping in blood clotting. Okay. Okay. So if there is a mutation in this gene, what will happen? Blood clotting, yeah, may not happen, and so bleeding will happen. So this is von Willebrand disease is associated with bleeding. It's a bleeding disorder. Okay. Okay. So you did this, which is very good. So let's go to next question. Okay. So this we were doing, and we encountered problem, but do it now afresh, and see if you are able to solve this problem or not. Question number 5. Okay, so first you have to go to Ensemble and UCSC genome browsers to visualize the human beta globin gene structure and obtain its nucleic acid and protein data from these sites. Can you go and do this? First go to Ensemble and obtain the DNA and protein data from Ensemble, and then go to UCSC genome browser and obtain the DNA and protein data from there. Okay, Ensemble is ENSEMBL. So could you go there? Okay, Ensemble. Yeah, beta globin, that's also okay, or you can just type HBB. No, these are different. Actually, go down. Actually, look for HBB, human beta globin. No, that's fine. Yeah, okay. Search. Okay, yeah, very good. So yeah, here it is at the top. Yes, this one. Uh, and then yeah, visualize the structure, which are these actually. So three exons. So these are the variants. So typically three exons you can see, but you want to download the data. So export data. Click on export data, and so you are able to do this. Ensemble. Yes, and you have to download data. So export data. Okay, so in sample you have to look for beta globin. So you can just type HBV. Yes, done. Okay, so did you do also UCSC? Okay, yeah, do a UCSC also, and HBB is at the top. And then export data, and then FASTA sequence, which is all good. And you want to, so question is, okay, nucleic acid, and okay, so you can include everything. Nucleic acid or cDNA coding sequence, and does it provide protein data? Yes, peptide sequence. Okay, and then get data. So next, and then you can click on compressed text. It will download to your computer. Okay, and now go to UCSC genome browser, and then HBB search, which is this one at the top. And for data, actually go to tools and then click on table browser. Okay, so on UCSC genome browser, you have to search for HBB, and then when you want to download the data, click on tools at the top, and then click on table browser. So here it is, table browser, and then it came pre-populated with the coordinate of this gene on chromosome 11, but you want to get sequence. So change the output format to sequence, because you want to get sequence of this gene. Okay, so are you doing UCSC genome browser to download the sequence? Yes. Okay. Okay, and then change the output format to sequence, and then click on get output. Okay, so okay, oh no, so you will have to change, because it all came pre-populated with, because we want to get the sequence of the gene, so you have to change the group to genes and gene predictions. Okay, so look at the top. It should be clade mammal, genome human, assembly HG38. It is about gene, so group should be genes and gene predictions. And if you want, you can change this to refseq, and it's okay. That is also okay. So NCBI refseq, and then position. So it came pre-populated with the coordinate of the beta globin gene, and then output format sequence, get output genomic. So it is not providing the protein, it is providing the genomic only. So that's fine. And sequence you want to include CDS exons, introns also. Okay, let's include everything other than promoter. So 5' UTR CDS, 3' UTR introns, and one faster record. Okay, this is okay. Send query to okay. Yeah, I don't have to send. You can get this from here. Yeah, you don't want to select this because we did this last time. Yeah, it is coming with all those options. Yeah, don't select galaxies for this. Don't have to. Then get output genomic, and then 5' UTR CDS, 3' UTR introns, one faster record exons, get sequence. So here it is. Okay, genomic sequence. Could you? Okay. Okay, then next question. We were attempting this question, but we are getting some error. So can you go and try this? Go to UCSC browser and use UCSC genome browser to get the summary statistics for genes. Okay, so this is something that I did. Can you do this? So summary statistics for genes in human chromosome 11. Okay, so now we have to get summary statistics for all genes. Actually, I had done this, but I want you to do. Okay, right. You did it. Okay. Yes.
In symbol,
Hmm.
it's human and HBV.
No, yeah, HBB actually, not G. Yes, this one.
And then go.
Go, yes.
Because this part I understood, but
Yeah.
this part I.
Oh, okay. So this one, first one, HBB. And then you can scroll down, you can see the structure of the gene, 3 exons, these blocks. You have to download the data, so export data. And then FASTA sequence, all this good, so go down and it will download all this CDS, protein and everything. So then next. Okay, so how many genes and their variants are there on human chromosome? So summary statistics for genes. So how many genes and their variants? How many? No, only on chromosome 11. Yeah, only, yeah, you see chromosome, so go to position, not genome. So position chromosome 11 and look up. When you will click on look up, you will get the first position and last position of this chromosome. So then, yeah, it doesn't matter even if it is, and then summary statistics, okay. 9,930, okay. Okay, and then do the next question. Okay, so this we were trying, but now I would like you to do on your own. So start with first UCSC browser and then use Galaxy to obtain coding SNPs in chromosome 11. Okay, try it. Yes. 20,000 in chromosome 11. Oh. Did you select ref6?
GIAN code.
Select ref6. Yeah, I don't know what is, if the question is saying ref6, then select ref6. So question is oh, summary statistics, then I think you are okay actually. But yeah, if you get option of ref6, then select ref6. Okay. So you all are trying the last part of question 5. Go to UCSC genome browser and then get the list of SNPs in chromosome 11, and then the list of coding exons in chromosome 11. Okay. Oh, you are still downloading. Okay. Okay. So good. And then this is good. Yeah, it should be bed format, because you no output format change from sequence to bed. Yes. And then okay, send to Galaxy. Oh. Hmm. Click on, yeah, click on this, and then okay, get output. Okay, so go ahead and keep doing. So okay, and whole gene. Oh, you, so coding exons, right? So you did for a SNP or not yet? Oh, if you are doing this first, and then you will do for a SNP, right? Okay, that's fine. Yeah, but make sure it's all genes. So delete that, and then look up. Uh, so then it is the entire chromosome.
Okay.
Yes. So entire chromosome actually. Okay. So you have to get SNPs in chromosome 11, and then coding exons in chromosome 11, and then you would intersect these two using Galaxy to get the SNPs that are in coding exons of chromosome 11. Okay. So first obtain the list of coding exons in chromosome 11 using UCSC browser and Galaxy. Then obtain the list of SNPs that are there in chromosome 11, and then intersect these two. Yes.
In symbol I come till here.
Oh, okay, good. So you will, for download, you will just download. It will download in your download. Yeah, it is here.
UCSC I came here. What is HBB?
Yeah, right, right. So you are here. So where is this? Oh, okay. Let me. Okay, I don't know what's happening. Okay, UCSC genome browser, and then type HBB.
HBB.
Yes, HBB, and then search, and then okay, maybe yeah, this no results. I don't know what's going on. Search results on. Okay, I will go to table browser. Table browser, and then I don't know what is all this actually. No, I think there is some problem. Genome is played. Let's go back. Mm, Genome browser. Thousands of for some reason actually. Let me.
This one.
beta globin, beta BATA, and then search. Search for beta globin and use the. Okay, I don't know. So let me do here and do with me. We should be getting the same result. So just close that window, and we will go to UCSC genome browser, and then so you are here, okay, and then type here HBB. Did you do? And then search. So I'm getting this actually. Okay, I'm getting this. You are not getting this. So search results on HG38. You are not getting this. Oh, I don't know. Can you? What is that called? Search history. Can you delete actually that? The cache. Just delete that from memory and then click on HBB here. So first you have to get to this. I don't know why you are not able to. So once you get this, then you should be able to. Which one?
By selecting the year, like 2022,
Hmm.
2030.
Then you get, okay, okay. Yes.
In this, am I supposed to get a table or sequence?
Yes,
I
yeah.
get sequences.
Oh no, you will get so do this.
In this one, it's getting
No,
I
it
get
is getting.
sequences.
Yeah, you no, I think some mistake happened. You will get not sequences, but show me actually what you get. Oh, so you are okay. You got this, and then tools, and then table browser. So here it is, and you want to get the sequence, right? This you are doing for sequence, or which part of the question you are trying to?
So last one, 11, gene
Which one?
chromosome 11.
Oh, list. Oh, okay, okay. Then I think you are okay. So yeah, but you have to have all chromosome 11. So delete this and look up. Look up. So here it is. The start and end. Okay. Okay, and you can do ref 6. Okay, and then you are interested in coding exons, so which I think you will send to Galaxy. That's fine. Bed. This is all get output. Coding exons, and this is good. Send to Galaxy. Okay. Done. Okay. Yeah. Yeah.
And Ahmad
Yeah.
might have also come, so
Yeah. Right. Right.
that
Yeah.
will be better.
That will be better. Right.
Next time we will plan.
Next time. Yeah. I think they could.
Colloquium. We will plan a colloquium.
Right. 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 Yeah. I think then.
And then we can block them. They can't
No,
go.
no, no. And we can then relax.
Then relax.
Okay. So able to do. What error you are getting? Okay. So yeah, let me try this again. Let's see. Position this Galaxy. Galaxy get output. Coding exams. So error is coming for coding exams. Okay, so I am doing actually, I'm repeating this. Yeah. So this is job number 6 to Galaxy, and let's see. Okay, let's see actually 5-10 minutes if we are able to do this. Otherwise, take this as your homework. So yeah, in the evening after your dinner, try this, and I will also try. Maybe we should be able to get this done at some other time. This could be server issue also, and it seems like some internet issue also here. So because of all this, we are not able to complete this. I'm repeating it, but I'm not getting any further from here. So send to Galaxy. But I have provided you the screenshots. So and I did this and I got the answers. So screenshots are there. So at some other time, just follow the steps in the screenshots. You should also be able to do it. Okay. Due to some technical problem, right now we are not able to, and that is something beyond our control. So we can't do much about it. But I have done it. I provided you a screenshot. So do it some later time, and you should be able to get it. Okay. Okay, then we will meet tomorrow at 10 or at 10:30. 10:30. Okay. 10. Oh, 10. Okay. Yes. वो इंटरसेक्ट नहीं हो रहा है वो अच्छा फिर से इसको कीजिएगा बाद में वो एरर विद इसमें लग रहा है ना कि एक क्योंकि सी व्यू you get it right you but what about view see the other one I want to okay this is yeah coding exons and show me snips snips yeah you got the snips so I think it should okay yeah it should actually so later on yeah it's simple actually this step is simpler than others so it should let me know tomorrow you are able to where is oh you are not able to get oh in both oh but you were able to get to step number 3 uh, the intersection step okay okay yeah try it some other time go through the screenshots you should be able to do it okay yes oh nothing yeah I think right just some other time so okay then we'll see you all tomorrow